Thank you very much. Last Monday, I gave a rather general presentation on biodiversity informatics and some general challenges and opportunities we have to demonstrate we can use information and communication technologies in dealing with the challenges of biodiversity conservation. Today's talk is rather focused. It's on one of those problems that I mentioned, particularly the problem of taxonomic identification or conducting large inventories. When I say large, I mean 8 million species that still remain to be discovered, described, and fully characterized using information technologies. Still, even though this is a very focused area, what I'm going to be presenting is a survey of some of the early and current attempts at making this cyber taxonomic work. That's one name that is given currently to the use of the cyberspace and information technologies to support the work that taxonomists have done for many years in a rather traditional manner, but that has been changing, I would say, over the last 10 to 15 days. And one of the main reasons why I picked this subject is, of course, that I'm working on this currently very profoundly, but also because I want to share with you the excitement of new developments that have happened over the last, I would say, 5 to 8 years that make information and communication technologies really very useful to tackle a specific problem, the general problem of fully automating or maybe semi-automating the process of identifying what is it that I have in my hands or what is it that I have surrounding me. In biodiversity-rich countries such as those in the Neotropics, Costa Rica being one of them, this is very critical because by far it is more than what we don't know than what we know. So I have divided this presentation in 5 topics. First, a brief introduction. Then, what are the 3 main early approaches to trying to automate or semi-automate the process of identifying species? 3 current approaches. 2 of them are very, I think, promising, and in fact, they are already delivering very exciting results. And then, even more focused, what is being done with respect to identifying plants based only on their leaves. There are good reasons why maybe leaves are a good way to start or to get, we still don't know how far, but certainly farther than expected. You know that in order to identify a plant, ideally you should have a flower, a leaf, a seed, have some sense of how it smells and some other information that would allow you to really identify it. Well, what can you do with scarcity of information? That's something that computer scientists deal with a lot. Given that I don't have all the information, how far can I get? And usually it's farther than experts expect. And then I want to wrap up with a few challenges and opportunities. I mentioned last Monday that the word biodiversity is used interchangeably with living nature. Even though biodiversity is, one can think of that as a number, it is really used as a synonym of living nature. But the official definition is this. This is what the CBD, the Convention on Biodiversity, says. It is the variability among living organisms from all sources, including, for example, terrestrial, marine, and other aquatic ecosystems, and the ecological complexes of which they are part. This includes diversity at the species level, between species, and also at the ecosystem level. We already talked about how many species there are estimated to live on the planet, 10 million. Macroorganisms, microorganisms, people don't even dare to say. And the need to organize this information with some type of classification scheme has been there forever. Normally, if you want to go to a systematic approach to any field of knowledge, you have to refer to Aristotle. And he himself, being such a wise man, he said, well, this is too complex. We should define a way of organizing nature, living nature. And he did it for 500 species. That's how complex the world was, or the biodiversity world was at the time, about 2,000 or more years ago. Even then, with 500 species, he said, we shouldn't simply put them in a single box. We should organize them somehow. And it took more than 18 centuries for Charles Linnaeus to come up with what is now called modern taxonomy. Modern taxonomy is more than 200 years old. He designed a system to name organisms, and it's called the binomial nomenclature. That's why we are all Homo sapiens and so on. There are Latin names given to all the organisms on the planet. And there's a very good reason why it's in Latin and not in French or Swedish. He was Swedish, or Spanish, or a quote-unquote living language. And this is the first two levels of many levels of the tree of life that describe how living nature is organized. Bacteria, we really don't deal with them quite often. Archaea, we don't either. It is the eukarya part that we normally think of when we think of living nature. Animals, fungi, and plants. The others are also there for a good reason. These are not the kingdoms, these are the domains. It's a more general concept that puts together all these organisms. And of course, I wouldn't dare to put here the 2 million species of organisms that are known so far, for the same reasons that Liliana Sancho yesterday mentioned when she talked about how to organize taxonomic trees and put them in a single screen. It's very tough, the problem of visualizing this information, and there's some interesting work being done with respect to that. So yeah, taxonomists set the stage for this process of trying to identify all organisms, but the workflows are very slow and error-prone, and also the expertise is in the hands of very few people, and they are one of the most endangered species, the taxonomists. This is called the global taxonomic impediment problem. How can we, with fewer taxonomists, still do the monumental task of identifying those 8 million species of macroorganisms that we don't know? And look, the sad thing is that we could say, well, at the pace that we have identified the first 2 million, it would take, I think it was about 500 years to do the whole thing. And that wouldn't be too bad a problem if we had not, we as human beings, affected the environment as we have. So we don't have 500 years to do that. The clock is ticking against us, and therefore we have to substantially change the way it is done. Now, this automated species identification has been a dream for many years, even before computers. Taxonomists were trying to see a way of more efficiently doing this. Another question, look, this presentation is about plant species identification. It's not about insects, it's not about fungi. Plants are very important. There are two reasons why plants are studied. One is because they are very, very important. When you look at the ecological level of biodiversity, you normally look at plants. You characterize an ecosystem more by the forest coverage than about what insects live there. Even though you should have a holistic approach, plants are very critical for those types of analysis. When we talk about payment for environmental services, most environmental services are provided by plants. So it's very important to be able to quickly conduct, for example, inventories of plants in a particularly endangered area. And also I have to confess, plants are easier than insects. Insects are three-dimensional. Plants are also three-dimensional, but leaves are very flat, and you can do some work with leaves, perhaps, and that's why actually we work with leaves initially. We know we have to work with other parts of the plants as well. 3 early approaches to doing this. One that even Linnaeus promoted was the single-axis keys, so-called single-axis keys. These are keys, these are in terms of programming, We could call them decision trees. It's a tree that has a question at each internal node, and by answering those questions at the end, you can say, okay, if you get to the really end, to the end of the path, then this is the species you are talking about. Or if you cannot answer all the questions, you may be able to say it's this set of species that match the answers to those questions. This was the earliest attempt at designing algorithms to identify organisms. There's a problem with this dichotomic or polytomic keys, and it is that taxonomists spend a lot of time making sure that the questions are asked in the right order. And by right order, I mean that the tree should be balanced. It should be as discriminative as possible from the very first question. You can have the same set of questions in different order, and you would end up with very different trees and with more inefficient ways of figuring out what is it that you have. But then, if the first question has to do with a flower, and you don't have a flower, you are doomed. You didn't go past the first question. And you have to do backtracking and some things that are undesirable. For those who are old enough to know Fortran and non-structured languages, I hope you see here, or you have a deja vu feeling with this. This is a key, a dichotomic key that is a good example of how it was done 200 years ago, 100 years ago, and last week in a publication that tells you how to identify some species of plants. It says, a dichotomous key for or using leaves. First question, compound or simple leaf? 1A, if it's compound, then go to step 2. If it's a simple leaf, which means the leaf is not divided into leaflets, then go to step 4. You all remember our old friend, the go-to statement, the famous go-to statement. It's there all over the place. But this is a linear way of describing it. This is an algorithm. This is a program. Of course, you can also draw it. It wouldn't fit in the book. You would need a piece of paper really, really big to have the drawing. But that's another way of doing it. The image, the figure is nice for human readers, but that textual description, that program, is nice for automated processing. An improvement on that is the multiple access keys. Here, they are called also interactive keys. Here, the user is in control, and this makes more sense with the aid of computers. Here, the user says, I have a flower and I have a leaf. Don't ask me about fruits, don't ask me about roots. I only have those two. And so ask me questions about the leaves and ask me questions about the fruits, and I'll see how far I can get. So in Costa Rica, we have about 11,000 species of plants. So originally, I know I have 11,000 candidates. And as I proceed with the questions and answers, I can get down to, let's say, 20. And I don't have information about seeds, and I don't have information about other stuff. So that's the best that the system can do, but maybe it can provide me with descriptions, images, pictures, access to the Encyclopedia of Life, or to GBIF, or to the National Herbarium. And from there, manually, I now have only 20 candidates, and I can do it the old-fashioned way. This is an example of Lucid, a language that is widely used. It's very simple. It simply creates a matrix. Each row is a character or an attribute, and each column is a species. So you put check marks there, and if you answer the questions in such a way that there's a column that has all the check marks, then that's your species. If we had to start from scratch, when I say we, I'm sorry, I mean computer scientists who have this algorithmic perspective on things. We would probably say, well, what if we measure organisms in all possible ways and then calculate the ratios of length and weight, and this is one way of reducing the problem of identifying an organism to a quantitative thing. Let's use some statistics, of course, because we know that not all organisms of the same species are the same size and weigh the same. Well, I have an example of a fish here. This has about 20 or so measures. This is the morphometric approach, in which you take those measures, and if you have 20 measures, that's just the beginning. This is the primary information. Quite often, the actual number or measure that discriminates is a ratio. The ratio of the length or the distance between the eyes and some other thing. Some of those ratios have been discovered and they are very useful. Some of them are not known, and it's perhaps with some data mining and some statistical techniques that we can figure out that in order to figure out if this is some species of fish, we have to look at the relationship between the size of the eyes and the length of the fin. It may be something as crazy as that, but it's just a matter of running all the data mining algorithms to see if such a thing is possible. Now, more recent approaches. This has happened, well, DNA barcoding at least in the last 10 years. And this has created a new field in taxonomy, which is called the molecular taxonomy. You look at the DNA of an organism, and of course, that's your fingerprint. Two problems. One, we would need to have a database of 2 million organisms. We still don't have that, but they are developing that database. And two, the genome of an organism is very, very, very large. Just think of having 2 million organisms, the DNA for each of them, which we still don't have, but let's assume we do. And now we have the DNA of a plant, and we have to compare it with 2 million. And for each of them, the amount of work is really big. The big thing about DNA barcoding is that biologists discovered that they have to look only at one gene, not the whole genome. And so they're creating that database. And for plants, 2 genes have to be looked at. So the complexity is a lot lower, but still the databases have to be created, and they're doing that. As you can imagine, traditional taxonomists felt threatened by this technology. I'm talking about 10 years ago. Still some think that this is not a good idea, and they're going to lose their jobs and so on. But now I have seen that there's more understanding of the complementarity of the two approaches. Both are important and needed. Crowdsourcing, I mentioned in this presentation, not because it's a quantitative approach, nor because it's an automated method. But it's an example of how information and communication technologies have been used to conduct inventories and to do identification of organisms. There are many citizen science efforts that allow people to take pictures of organisms, upload them, and then there's a community of empirical experts, or professionals who help identifying that. And the explosion of information nowadays is really big. GBIF, which didn't have a single picture of an organism 10 years ago, now has more than 1 million pictures. iNaturalist is an excellent example of this approach, and PlantNet, about which I will talk a little bit more in a moment too. A second approach, which is a third approach, I should say, which is the focus of my work and today's presentation, is image-based automatic identification. You know that for many years, computer vision has been used very successfully in medical imaging, optical character recognition, and also in biometrics, but only recently has it been used with organisms. And they started, not surprisingly, with butterflies, because like leaves, they're very flat, and because some families, not all, but some families can be fully identified, the species of those families, by looking at the patterns of the wings of a butterfly. And there are many machine learning techniques, SURF, SVM, neural networks, LBP, that have been successfully used. There has been a revolution, a second coming of the neural networks and some artificial intelligence techniques, thanks to a better understanding of how they work, and also the computing power that is now available. How does it work? Well, there's supervised and non-supervised learning. I'm going to talk only about supervised learning. You first take images of different species, hopefully many, many images. Many means at least 20 images for each species. You put them in a database, and then you have algorithms that clean them, that segment them. That means the leaf bits or pixels are separated from the non-leaf pixels if it's a leaf that you are using. And then the relevant features are extracted. The relevant features for a leaf could be the silhouette, the texture, some morphometrics, or it could be also the veins, which are very important, and some spots. So what is one of the first key things to be done? Move it from the bitmap domain to say, a histogram domain, and then use that to train the data set. Look at this very simple problem. Suppose you have taken pictures of leaves that are all, let's say, looking up north. The apex is up in the middle, and the base is at the bottom. And you have all your images like that. You don't want really users to take all pictures so that they are in that same position. You would like those pictures to be rotation invariant. You just give me a picture, an aerial picture of the leaf, the back and the front, or just the back or just the front, in any position. That's what you want. So if we have an image in this position and the user took the picture in this position, he or she will probably have to try 360 different degrees of rotation until they really match. We don't want to do that. There are ways of transforming those bitmaps into more summarized representations. You lose information, but you get the most important information, and those are rotation invariant. So you don't have to have a lot of information, and you don't have to have a lot of processing. So once you have that database D, you train an algorithm with that data set, and then once you have a trained algorithm, you tell the user, give me an image, and I will analyze that image, comparing it with the processed versions of the images, not with the bitmaps, but with the histograms or whatever they have. One nice name, it's still too ambitious, that the French researchers from PlantNet used to describe their app, because it's a mobile app, is that they want it to be the Shazam, if you're familiar with Shazam, the Shazam for plants. Just as when you want to identify a song and you use Shazam. With leaves, I will simply mention that we know that leaves are not enough to fully identify species. But there are two reasons why we want to work with leaves. One is because they are part of most keys. So they are very important to identify species. Second, botanists didn't take full advantage of the information that leaves have. And the reason is they always had flowers, they always had seeds, they always had some additional information. So they didn't care about making the best out of the information that you can get out of leaves. And thirdly, I should say that what we expect is that the algorithms we use for leaves can also be used later for flowers and for other components of the plants. Now, the big player nowadays is deep learning, convolutional neural networks, and they have done really very, very interesting, amazing stuff. They have neural networks with more than 100 layers. That's a lot. Usually neural networks have 5 or 6 layers. 100, 120 is just outrageous, or was outrageous before. Now you can have that type of thing. There are two limitations, however. One is that neural networks require a large amount of images. So for each species, you probably want to have 100 pictures or 200 pictures. Multiply that times 14,000 in the case of Costa Rica. And also that they have a black box behavior. If I show this to a taxonomist and I say, well, my system says this is this species, he or she will probably say, why? Can the system explain to me why the system came with that conclusion? And I would probably say, well, because it says it is 99.99999% possible that it is that. And I have run it 20 times, and that's the number it gives me. It's almost 100% certain. It would be good for any expert system, even the old-fashioned expert systems. It is a requirement that these systems have an explanation because it allows developers to polish the piece of software, and also because it gives more confidence to the users. All right. I haven't seen the 1 minute card, but I suppose it's over already. So let me take this final minute just to remind you of a couple of challenges. One is the taxonomic impediment for such a huge amount of information, and the other one is that we want to automate the species identification, not just to develop this encyclopedia of all species on the planet, but for practical reasons. For example, control of trade of endangered species. It would be nice if you had an application that allowed the officials or the officers in charge of that to have an automated identification, and also to conduct inventories rapidly. Last slide. These are 3 examples of interesting opportunities. The Global Biodiversity Information Facility, Encyclopedia of Life, TADWIC, iNaturalist. These global initiatives are already working together. They have connecting channels of information, and that's why GBIF currently has more than 1 million images. Most of them come from iNaturalist and Encyclopedia of Life. Wikipedia is providing images to all of these initiatives as well. So that combination plus crowdsourcing has made it possible to talk now about a global database. PlantNet, this Shazam of plants, they have done it already at the European level, and they are promoting that we do it in other parts. I have a very bright doctoral student spending a semester with them in Montpellier precisely to work on this. Now, herbaria. Herbaria have invested a lot of time, a lot of money in creating their treasures. I call them treasures, where they have sheets where they mount plants with all the elements necessary to help identifying other plants. It's a lot of time, it's a lot of effort, and they are only starting to digitize that information. It would be very nice for them, because their budgets have been cut substantially over the last 10 years, that's part of the bad news, to process those images so that we can extract the elements. It's not a trivial task, it's hard. Take those images, get the flowers, get the leaves, get the fruits, get everything in there separately so that we can feed this global database that needs so many images to really be reliable. And finally, with respect to deep learning, I cannot emphasize enough how important they have become, because among other things, they allow you something magical. And it's because it's a black box that people can only say it's magical. They allow you to take pictures of parts, elements of a plant, or the whole thing, and feed that to the database, the training database. And with that, those are very, very, very different images. You can come with a picture of a flower, a picture of a leaf, a picture of an element, or a picture of the whole tree, and it can give you a relatively good rate of accuracy in the identification. They are at the 70–75% accuracy rate with small sets of 1,000 species and so on. Other approaches have 90% or more accuracy, but they require pictures of just flowers or just leaves, for example. Thank you very much.
Thank you.
Sorry about the time.